Hey guys, in this video we're going to go through the first two sections of chapter 16 on gene expression. So we'll talk about what gene expression is, and then we'll go over prokaryotes because they're the easiest to learn first. So when we talk about gene expression, all the different cells in our body have the same genes if we're talking about somatic cells. Of course, regular body cells versus gamete, sex cells are not going to have the same DNA, right? Because we talked about those are going to be a little bit different. But our normal, regular somatic body cells that are going to have 46 chromosomes in them, every cell, all of them have the 46 chromosomes and they all have the same DNA. But every cell doesn't look different, right? The cells that make up our eyeball are not expressing the same genes as the cells that make up our liver and gallbladder. Every cell expresses genes a little bit differently. So why do they express things differently? They're going to have different needs, and they will turn on the genes that they need to perform the jobs that they need to do versus the other cells that don't need to do that. So if we were talking about, I don't know, the pancreas, for example, the pancreas is responsible for producing insulin. I don't need my eyeball to produce insulin, but I do need my pancreas to produce insulin. And we've got the same genes to make insulin in both cells located in all over the body, but only this guy is going to turn on the insulin genes to make those. So do we want to express every single gene that we have all the time, 24/7? No. If we did that, that would be a complete waste of energy. It would be a waste of time. The DNA, every time we unwind it to express itself, takes up more space than if it's tightly packed in the chromatin that we talked about before in its chromosome. If we had to express every single gene, it would take up way too much space. It would not be efficient. It would be a waste of energy. So we only express those things that we need to express. We are constantly expressing like daily maintenance issues that the cell has to overcome, and then we will express other things on an as needed basis. We will turn on those genes for gene expression. So when we compare these guys, there's some differences. Overall, the basics are the same. Gene expression is going to be central dogma. We're going to go from DNA to RNA to protein. DNA to RNA is transcription, where you're writing your message. RNA to proteins are going to be translation, where you translate that mRNA into amino acid proteins. Where this is happening is different though. So when we talk about prokaryotes, remember we said that prokaryotes didn't have a membrane-bound cytoplasm, that this is happening, the DNA is just hanging out, membrane-bound cytoplasm, membrane-bound nucleus, that the DNA is hanging out in the cytoplasm. It's not stuck inside of a membrane, so it's in the nucleoid region. So we can transcribe and translate pretty much at the same time. So transcription and translation is happening in the exact same location, pretty much at the same time. We're going to transcribe, of course, before we translate, but it can happen really quickly, within seconds. This is happening. So how can we regulate this when it's all happening at the same time? We don't. We regulate at the transcription level if you're a prokaryote. If you're a eukaryote, we have the membrane-bound nucleus that we talked about before. So this is going to be where our transcription happens inside the nucleus, and that is a totally separate event. We're going to unwind the DNA, we're going to transcribe our messenger RNA, and we talk about all the different processes that it goes from rough draft to final draft, and then it finally leaves the nuclear membrane, goes out this pore, goes into the cytoplasm, and hits up with the ribosome so it can be translated into our protein. So these are two separate events. So we can regulate every step along the way as far as gene expression, and we'll go into those in the next video. But there is epigenetic, transcription at the actual transcription like this one, post-transcription, because we said rough draft to final draft, translation, taking that messenger and translating it into the amino acid, as well as post-translation. So there's a lot of different places on eukaryotes where we can mess with the gene expression and stop it if we needed to. So eukaryotes are a little complicated. Prokaryotes, a little more basic. And so in this video, we'll go through the prokaryotes first. A neat video that came out last year, the year before, was this one on the astronauts. And it is two twins, and I can't remember their names. I think one of them's name is Scott. And one of them spent almost a year at the International Space Center. And the other twin, who's also an astronaut, they are identical twins, the other twin stayed here on Earth. And then they came back and they compared the one who went out on to, who stayed in the space station and seen and looked and see what genes is he turning on, what genes is he expressing that he needed to express out in space versus his brother that didn't need to express those genes, and then they called them space genes. So it's kind of a neat video explaining it in generalistic terms, but I thought it was pretty cool to be able to compare two people, identical twin brothers, and we can compare them, what happens with space and what doesn't. How does your gene expression change depending on what you're exposed to? It will change depending on what you're exposed to. So it was neat. Moving on. Table form on comparing prokaryotes to eukaryotes. So again, no membrane-bound nucleus in a prokaryote, just a nucleoid. DNA is hanging out in the cytoplasm, not in the nucleus. They are almost simultaneous for transcription and translation, and we are only pretty much able to, prokaryotes can regulate their gene expression at the transcriptional level versus all the other places for eukaryotes, epigenetic, transcription, blah, 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 all the places that eukaryotes can regulate expression, and we're gonna get to those. They have a nucleus, it's DNA stuck inside the nucleus, and RNA transcription is happening inside the nucleus. RNA translation is going to happen out in the cytoplasm. So two different locations, two different processes that are not happening at the same time, like a prokaryote. Let's move on to the next section and actually talk about how gene expression happens. So in a prokaryote, we cover the basics. So there is the operon. An operon is going to have three basic parts to it, but what it does is it's going to code for that protein that you need to express in that cell. It's going to cover that protein or even a group of proteins that would be as part of a pathway. There's three types. There is a repressor, there's an activator, and there's an inducer. A repressor, I'm stopping this from happening. I'm repressing it. I'm keeping it down so it can't come back up. Repressors are inhibitors. Activators are just like they sound. They're going to turn it on. And then inducers have to meet certain qualifications. You have to check certain boxes before they can turn on. This and has, we'll get into it. We'll get into it. These are the three different ones: repressors, activators, inducers. On an operon, there's three basic parts. You're going to have all of this green stuff here are going to be the genes that you want to code for, that will code for the amino acids later when it goes into getting translated. Before you get to the genes, you're going to have a promoter, and then there's another place called the operator. Promoters versus operators. Promoter is going to be where this guy is going to attach on. Your RNA pole is going to have the promoter region of DNA that it is looking for. If we are talking about prokaryotes versus eukaryotes, we both have this region here, the Tata box, and that's going to be a place where it's like the landing strip for RNA polymerase. It's looking for that place so it can bind on, and that area of the DNA strand is called the promoter, and RNA pole is going to bind on to the promoter at those locations. Then we have the operator. Later when we get to activators are going to help with the promoter. Operators are going to be where repressors are, so they can't get to the gene section later on. We won't talk about the factors or anything, but there's numbers. You see in this picture, there's numbers. So here's our transcription site is right here where we switch from the operator to the genes. Anything past the transcription site when we're in the genetic material is going to have a plus sign. So all the nucleotides after this location is a plus sign. The nucleotides behind that location is a negative sign. So we can see the Tata box is -10, and this repeating, this TTGACG region is -35. So we've gone back 35 spaces from our transcription. So these are downstream, and this area is upstream, and it all depends on here at the transcription start site. If you're past that, you're downstream. If you're up above earlier, before we get to the transcription site, then you would be upstream. So the three different types of operons. One of them is repressor, then we got an activator, and then we got an inducible. So TRIP is tryptophan, have an amendment. TRIP is tryptophan, and it is a repressible operon. So we said that you don't want to turn on a gene and express a gene and go through the effort of making that protein if you don't need to. It's a waste. If tryptophan is in the environment, why would I turn on the bacterial genes to make tryptophan when I don't have to? It's already all around me and I can just use that stuff. Why would I make my body have to synthesize tryptophan if I didn't have to? So in this case, it's a repressible operon. So we have a repressor and it's going to attach on at the operator portion of the operon. And the repressor is going to, here's our RNA pole that's going to lay down the RNA. It's going to come across through this DNA and lay it down, but it can't. It is blocked by the repressor. The repressor only sticks to this operon because it has tryptophan. The tryptophan is going to, tryptophan in the environment is going to stick onto this repressor and they will block the RNA pole so it cannot make the message. It can't transcribe this message coding for making this protein when we already have the protein out there. That would not make any sense. It wouldn't be efficient. If we don't have tryptophan in the environment, if we haven't absorbed it, it's not out there, then we need to make it. So if it's not there, then we have nothing in this area right here on the repressor. The repressor won't bind, and if it's not bound, then RNA pole can just chug along like a train track and go on down the operon and transcribe those genes into a messenger RNA. So tryptophan acts as our repressor to turn off gene expression. So this is a repressible operon. Tryptophan is an example of a repressible operon. It is a negative regulator. What about the opposite? What if we wanted to turn something on? Bacteria, prokaryotes, are able to utilize different types of sugars. So can we. They can utilize different types of sugar, and if one thing gets low, say glucose gets low, if glucose levels get low within the cell, there's another substance that starts to accumulate as glucose levels get low. So when glucose levels drop, this guy, our catabolic cyclic AMP, is going to start accumulating. Cyclic AMP is going to bind with, here's cyclic AMP, it's going to bind with our catabolic activator protein. So here's catabolic activator. Our CAP, catabolic activator protein, will bind with cyclic AMP that only accumulates when when glucose is low. Glucose gets low, cyclic AMP rises, cyclic AMP is going to bind with the CAP, and now that is our activator. Because now this is going to, just like it says, it's a catabolite, it's going to help us metabolize other forms of energy. So if glucose isn't around, maybe another sugar source is around and we can break down that sugar source. So this is an activator. Glucose drops, cyclic AMP accumulates. If we have enough cyclic AMP, it's going to bind together with the catabolic activator protein, and this is an activator. It is meeting up with RNA pole, and it's going to turn it on, and it will go down and run through the gene expression. So this is activation with CAP. Inducible is our third type of operon. So this is going to have certain specifications, boxes that it needs to check for gene expression to happen. So it's not just an on or an off. We have to make sure certain parameters are happening. So when certain parameters are happening, we said that, say glucose gets low. If glucose is low, we just talked about what accumulates. Cyclic AMP accumulates. And cyclic AMP can bind with the catabolic activating protein. So this happens when glucose levels are low. Glucose is low, and we start accumulating cyclic AMP, binds with CAP, binds with the RNA pole, and activates RNA pole. So we're going to activate RNA pol, but what is it going to express? What genes is it coding for to express? What are we trying to do once we turn it on? Well, in this case, this lack operon is inducible because not only does glucose need to be low to turn it on, but we also have a repressor. So if we have the ability to, if we have the ability to break down glucose and we've got plenty of glucose, great. If glucose drops, we're going to look for other types of sugar. Another type of sugar is lactose. So if lactose is in the environment and glucose levels are low, then let's try and break down lactose and use that as our energy source. If lactose is not around, like here, there's no lactose. If there's no lactose, why would I waste energy expressing genes to try and break down lactose if there's no lactose to break down? So here we have no lactose, the repressor stays in place, and we're not going to undergo gene expression. In this case, we do have lactose. So when we have lactose, we want RNA pol to go down and start expressing the genes to break down lactose, and we can use that energy source. We can do this without glucose levels dropping, but it runs faster when glucose levels are lower because it has that activation that happens when cyclic AMP comes along and binds with the CAP and activates RNA pol. So we have to have these conditions met. There has to be lactose present. We must have lactose present. And we must have low glucose levels that will lead us to this step here and activate the RNA pol to turn it on and send it on down its way. So this is an inducible. Glucose has to be limited and lactose has to be present to serve as the fuel source. Otherwise, why create the ability to break it down if it's not there to break down in the first place? So here's the table that tells us the conditions needed for the lac operon that breaks down lactose to work. And we said if glucose is present in the environment, then why would we worry about lactose if we can break down glucose? So if glucose is present, no, we don't really need to. If glucose is present and lactose is present, then we could do a little bit of it. We've got glucose, it's our favorite place, but we could also use lactose. And we could do a little bit of gene expression to break down lactose because lactose is present. We could break it down too. So we'll do it a little bit, some. If glucose is not present, cyclic AMP is going to bind with the CAP and we will have gene expression. But gene expression won't happen if we're missing lactose because remember, lactose has to pull the repressor off. If lactose isn't there to pull the repressor off, we don't get gene expression. So we must have low glucose. Cyclic AMP needs to bind with the CAP. And lactose has to be around, otherwise we have nothing to break down when we express these genes. And so now, yes, this is what we want for this type of gene expression. So summarizing, there are three. There is a repressor, like tryptophan. There is an activator, like the cyclic AMP, the catabolic activating protein, the CAP. And then there's an inducible that has certain conditions that have to be met before it will turn on. So we'll cover eukaryotes and how they are a little more complicated in the next video.